An adventure. Right. So this is going to be a quick 5-minute update. Kim, you're looking like you're amused. Am I being heard? Okay.
Nope.
We'll hold the signs up in front of the camera on our end, or maybe just shout at you.
Yeah.
Go for it.
Okay. So a 5-minute update on using acoustic telemetry to project species distributions in Chesapeake Bay as a result of climate change. This is a new project that we're starting. I'm actually pinch hitting this talk for Bethany Williams, who's the PI on it, but she got pulled away for a boat course. So she's probably out there having fun somewhere on the water. Could you change the next slide? Let's see what she has for me here. Right. So this is some work that is funded through the Climate Ecosystems and Fisheries Initiative. This was part of the, the Inflation Reduction Act, I think. Big budget, multi-agency thing going on here. A lot of fisheries components, a lot of climate and weather components. For our tiny slice of this, what we're proposing to do is exemplified here in this study by Creer et al. From a few years ago, where basically we're going to use acoustic telemetry to develop current species distribution models throughout the bay. And then once we have a good understanding of what the variables are that are driving those species distributions and habitat suitabilities, we're going to pair the model with future climate conditions to look at, what is the distribution going to be like in 50 years or 100 years, and depending on different emissions and climate scenarios. So that's kind of the big picture of what we're after, with lots of details to be figured out. We're just at the very beginning of this project. We've talked to a few folks there in the room already about it, but we're definitely looking to interact with more people, answer other questions that people have, and also get a couple of aspects of feedback from you guys. And I'll touch on those here in a second, I think. What's the next slide? Yeah, so we are right now with the funding that we have, we're looking at trying to tag maybe 50 to 100 individuals of a few different species. Pretty sure we're going to do striped bass and red drum. Both of those are going to be moving around in the bay and probably do some interesting things in terms of their migration patterns, and maybe changing things seasonally and interannually, and doing things different as the environmental conditions in the bay change, maybe the timing of their migrations. If there's another species that you're thinking, hey, I wish those guys would look at this one, let us know. Like I said, we're at the very beginning of this process. And so if there's another species that is particularly important to you, either from an academic standpoint or a management standpoint, love to have a chat with you about it. And maybe you're tagging those. This might be an opportunity to get another 100 tags out there. Right now, we're looking at using V9s for a couple of years to hopefully maybe get a little bit of that out migration and in migration stuff as things change from one year to the next. What's the next slide? Right, I leaned in when I heard people talking about this. This is sort of the current distribution of maybe just the last 2 years of receivers that are out in Chesapeake Bay. And we're really looking to take advantage of this to make this particular study work. It only works with a network of folks, and having that wide distribution of receivers out there is what we're counting on, but we're not freeloaders. So we are going to purchase another, I think we purchased 25 receivers, and those stars that Kim just put up on there, those are hypothetical where we might throw them. But we want to fill in a couple spots, that backbone array that another part of NOAA manages. I think it's the blue, the purple, the darker purple, and the pink down there at the bottom. We're looking to sort of fill in that line right there across, what is that, the Rappahannock, where the shipping channel kind of goes diagonally up through the bay. Looking to add a line of receivers there, sort of a holy gate, if you will. It's not going to be, a double-decker gate or anything like that, but it should should detect some things moving up and down the bay. And then, you know, at the mouths of other promontories and other major rivers, some on the eastern shore, there's kind of a gap in receiver coverage. Again, these stars are just hypothetical, and we'd love to hear from other folks about where some key places might be that they'd be interested in seeing a receiver. If we can put one there to help you in your study, and it works for us too. We'd be glad to do it. So just reach out to us and let us know if there are certain places that you'd be really interested in seeing an additional set of receivers placed. Let's see, I think we covered all that. Yeah, the time period for this study is going to be, you know, kind of over the next 4 years. 2 summers of tagging, so looking at getting, you know, maybe 300 or so tags out in those species that I mentioned, and maybe others. Every 6 months, we'll do some receiver downloads, and then probably trying to look at getting our deliverables and, you know, results in the 2028 kind of time frame. I hope that's the last one because I don't know that I have anything else to say. Yeah, great. Yeah, so if you do have comments, questions, you want to just know more about the study, or you want to talk a little bit about a good spot to put some additional receiver assets that maybe is a little bit lacking in you know, some aspect for one of your studies, be glad to hear about it and tell you more about it. Reach out to Bethany or me, and we can talk. Thank you.
Thanks, Matt.
Thanks, Matt. All right. So for all the other speakers who are in the room, you'll get the cards for 3 minutes and 1 minute left. We can do one quick question for changeovers, and if there's any time left over, we can allow other questions. But our first speaker in the room is Rachel Rodet, one of our Student Travel Award recipients, presenting on her work at University of Delaware.
Hi everyone, I'm Rachel Rodet. I'm a graduate student at UD. Like Matt mentioned, I just want to thank ACT and Maricus really quickly for selecting me to be one of the recipients of the Student Travel Awards. I'll have some further updates at AFS Hawaii, so if you're going to that, come check out my talk. So today I'm going to be talking to you about American shad in northern Delaware. First off, American shad are a diadromous fish species that are native to the east coast of the US. They're a coastal migratory species. Thanks again, Matt, for introducing the Alosas. And they experienced a dramatic population collapse in the early 1900s due to 3 prevailing reasons: historical overfishing, degraded water quality and pollution, and also physical impediments to spawning grounds such as dams and weirs and other habitat loss. So we can see the figure on the right shows catch decreasing since the 1900s along the Atlantic coast and never really recovering. Going to focus in on the Delaware River because I go to UD, and we can see that catch also decreases exponentially since the early 1900s, and there's no recovery. The Delaware River is the longest undammed river east of the Mississippi and has over 200 tributaries, which contain hundreds of dams, lowhead dams, that are mostly obsolete or abandoned. The Delaware River is about 500 kilometers long and becomes non-tidal about halfway through in Trenton, New Jersey. So this is what the Delaware River watershed looks like since it's a little bit slept on. And I'm going to zoom in into the Brandywine-Christina watershed that is in southeastern Pennsylvania and northern Delaware. The Brandywine-Christina watershed is made up of 4 rivers: the Brandywine River, Christina River, and Red Clay Creek and White Clay Creek. So in these 4 different rivers, there are over 35 low-head dams. And then I'm going to zoom in even more into the city of Wilmington in northern Delaware. And this is showing the first 11 dams on the Brandywine River in northern Delaware alone. So this is looking at Newcastle County. And dam number 1 in Wilmington, Delaware was removed in 2019, which allowed us to ask the question: are American shad using this newly available but historic habitat they haven't had access to in over 200 years? And what are any behavioral impacts due to acoustic tagging? So, sorry, this isn't a fancy map, but we deployed 3 different receivers in the Brandywine River, one above the removed dam, which is the red line, that's referred to as Brandywine 1, and then 2 downstream of that. Since this, we've also deployed receivers in the other tributaries of the Brandywine Christina watershed, but I'm just going to focus on the Brandywine River. And then we also relied on over 60 receivers deployed by Delaware State University's Dwayne Fox and DENREC. So over the last 4 years, we've tagged 44 American shad. So we're looking at low sample sizes compared to what you're used to. And this year was pretty bad spawning run. So we're actually going to switch species for the rest of our season and start tagging predators of American shad, looking at blue cats and really, really big channel cats. So once again, visit my talk at AFS for updates. And I'm just going to focus my analysis for the remaining 2 minutes on 2021 and 2022. Shad were measured for fork length, total length, and sex to the best ability of the researcher. 5 fish were removed from analysis due to latent mortality post-tagging or fewer than 5 detections. And then we used the detection events function in the GLaDOS package to calculate occupancy periods. And what we found was that American shad preferentially use that habitat above removed dam number 1. So within all of the systems, American shad are spending most of their time within the Brandywine River. That's 72% of their time. We have individual fish along the x-axis and then a proportional occupancy from 0 to 100% along the y-axis. And the prevailing color here is purple, which is the Brandywine. And then when we break this down into the 3 receivers that we have in the Brandywine, so Brandywine 1, 2, and 3, we see mostly green. They're spending most of their time above that removed dam. And then we looked at some different behaviors. We found a couple that overwintered in the Atlantic Ocean and actually made their way up to Nova Scotia and back to the Brandywine, exhibiting high site fidelity. A few that exited and then returned to the Brandywine within that spawning season. A few that initiated fallback behaviors immediately after tagging, and then some that remained within the Brandywine for weeks to months after tagging. That was about half of our fish that remained within that one river for their entire spawning season. So, are American shad using this newly available but historic habitat? Yes, and they're preferentially using it. And then what are any impacts due to acoustic tagging? 2 fish performed fallback, 3 had tagging related mortalities for about 15% having some kind of handling response. And with that, I'd like to thank all of my funders and collaborators, and I'll take one question, I guess. No, I like that.
Thank you. All right, our second speaker is Dave Seaforth from the University of Maryland Center for Environmental Science.
Oh, I gotta go here. So I'm going to talk about work on a very small system, tributary of the Nanticoke called the Marshy Oak Creek. This is maybe the smallest system in which Atlantic sturgeon spawn. I guess I can introduce the surge to sturge. This was when you capture a fish with DNR, they let you name it. So this is named after Serge Doroshoff, who was a famous expat from the Soviet Union, became a sturgeon expert at UC Davis. All right, let's see. There we go. So we've been working on this for 10, 11 years with DNR, and it's been one surprise after another, and it's all related to telemetry data. I've been working on sturgeon 30 years, and they just keep fooling me time and time again. And so, of course, first they were spawning in fall. Here's a fish that, and I should advertise Nicholas Coleman, another ACT recipient, has moved on to great career change. He's at the Southeast Fisheries Science Center. And Joy, you may be interacting with him. He's the offshore wind staff member at Miami right now. And then finally, this thing that sturgeon go way up high. I always thought sturgeon were big river fish, but no, they're going up into, wadable parts of tidal creeks, way up to head of tide. I want to advertise that this work really has led to important conservation management outcomes. So we should do a write-up perhaps for a couple different things, but one is that the telemetry science we did, we put that ahead of public, put it in front of regulators. We made a real stink, and Mike helped me with this. Here's a nice animation. And what we were trying to do was to make people aware that a sighting for a very large RAS, no, you gotta help me, thanks. It's gonna be jumpy. No, oh well, that's okay. Those dots are going to move up towards Federalsburg. But in any case, we had direct telemetry data that said that sturgeon were moving up into these really narrow parts of Federalsburg. And this was a sighting location for a really ridiculously large salmon factory, land-based salmon factory. And you'll hear about these coming online, but this was just a poorly sighted one. And so we had the telemetry data in hand that really helped thwart that. Well, the other thing we had was Nicholas and his thesis work. And so Nicholas figured out run size, and he did this through telemetry and combining it with side scan sonar, and came up with a very small run size. So because this is a small system, it's the Marshy Hope Creek, the Nanticoke, and the lower Nanticoke would then exit into the Chesapeake on the eastern shore. But we had side scan sonars, and sturgeon are very big reflective objects in terms of acoustic cameras. So you can see the image there. So we could go through and run replicate passes, and this is work done by University of Delaware, John Matson, and we could get counts. So we could count all the sturgeon in the river, but then we don't know if they're staying put or not, right? And we don't know if they've been there for a while and those kinds of things. And that's where the telemetry helps. So we can integrate telemetry to know the comings and goings of sturgeon, different parts of the spawning reach, and that integrating them can allow us to come up with robust run size estimates. So what you see in the red dots is basically we're accumulating sturgeon as they're coming in and then they're leaving. So it gives that dome shape with date. If we just look at the cumulative profile, that when it peaks, that gives us our estimate of spawning run size. So we have very low spawning run sizes in both years of study, 32 and 2020, and about double that number. We're still scratching our heads about why it changed so much. But one idea is that we might have had a better census that year in terms of covering the spawning season, which is going to vary year to year. All right, well, I'm all done. But one of the other outcomes here is that we don't have a recovery plan. It kind of pains me to say that. We have recovery plans for green sturgeon, for pallid sturgeon, for whites, well, some populations of white sturgeon, but nothing for Atlantic sturgeon. That's not good given how long it's been listed. So one of the things that is important in recovery plans are things like targets, thresholds. So I would suggest we're getting increasing numbers of spawning runs across different systems. You can see them here. Two of those systems, we got those through combining telemetry and side scan sonar approaches. Thanks.
Anyone have a question for Dave? All right. No questions? Let's see. Which one are you? Yeah.
You told me.
All right.
Sorry about that.
Yeah,
Get
so
some
the
up here
next one-
alone.
Yeah, so the next one is Rob
Di Giovanni, it's okay.
Giovanni, Tom Twister,
Sorry.
and Sarah Callan from Mystic Aquarium and.
Atlantic Marine Conservation Society.
Yes, excellent.
Glad I got that right. That would be embarrassing. So, well, as we try to load this, very much for having us, and you'll see why you don't invite us back after our not being as prepared as we should be. But to our credit, we just found out we were going to be trying to put slides together. So, Sarah, do you want to introduce what we're doing first, or do you want me to keep? Because I'll just keep talking forever.
I'll start. But yeah, my name is Sarah. I manage our animal rescue program at Mystic Aquarium, and we've been working very closely with Rob and then the team at Atlantic Shark Institute and Rhode Island DEM on a new project that we're really excited about, which really started And I guess for our animal rescue program, I will also share that we respond to live or dead marine mammals and sea turtles, and Block Island and Rhode Island are part of our coverage area for the aquarium. And so this whole project kind of started because we have a growing seal population around Block Island, and there's been a lot of interest in the seal shark relationship in that area. Right now, it seems like sharks are kind of bypassing the south side of Block Island from Long Island, or they're going straight to Cape Cod. But the question kind of came about, what's the seal population going to take around the Block Island area for it to be worth it for the sharks to either take up residency or make the stop to the north side of the island. So that's kind of how this project all in collaboration all started, and it's really kind of progressed since then. So we're really excited.
So, yeah, I think that this is really the epitome of how the project started. We heard about animals over here. I've been flying, doing aerial surveys. Short after Payne and Seltzer did their work in '89, looking at seals, which are the black dots there. We started looking at haul out sites for seals in southern New England and New York. So we started to see these seal haul out sites popping up. And I've flown the north part of Block Island for many, many years because it's always great to survey and not look at anything. So you can say that when something happens, they're showing up. And it turns out that for a good portion of the time, I missed all the animals that Sarah was seeing. And then we talked about it and she goes, hey, I'm seeing them on the northern tip. So this project kind of evolved to where you're seeing a couple of animals to seeing a large number. And we're also seeing now more seals haul out sites in the last couple of years, even in New York on Gardner's Island. We have them on the northern tip, which almost looks the same as this haul out site. Fort Tyler, which is just north of that. So there's a lot of changes there. And so this project, as it started to look at the movement of gray seals and harbor seals and harp seals after they're rehabilitated and released, to look at the success of that. Also to look at, are they staying around Block Island? There's an array that's put out there by the Atlantic Shark Institute. So we kind of built some things together. This work is authorized by our partnership with Marine Mammal Health and Stranding Response Program. And then I have a research permit under U.S. Fish and Wildlife to do some sea turtle work as well. So what we did is we decided this would be a good idea. And I can't read. So a lot on this slide, but what we're really trying to point out is how do we actually make this animal give us a lot of information. So you can see up in the top left, we have the satellite tag that we'll use. We have, we use epoxy to put that on. And we have the flipper tag that we make up to put the acoustic tag on that animal. And then we have a regular flipper tags. So then we set that up, put the animals back out into the wild. They're definitely happy about this. But the interesting part is you really don't notice the flipper tags much at all. And that's what I always find interesting is we put them out. We don't get a lot of tag returns, but this might get us some tag returns. The cool part about this is I've been sitting in a room with you guys for 2 days and I've been watching where all your arrays are. And, you know, at some point, a picture's worth 1,000 words. And I looked at, these are all the last 11 animals we put out. Obviously, we didn't clean too much of these data up while I was sitting back there before. But we can see that the yellow dots are usually data. Dan, you said it's more art and science involved in this. Well, those are the dots that we would wonder if they're accurate positions, and we would maybe tease them out of what's going on. But from Keith's work, you can see that he has arrays up there. So we might actually be hearing these animals as they move up and down the coast. And then the graph on the right is showing us an animal that was up in Beverly. It was a gray seal. It was captured, brought down to Mystic. It was released. And the movement is what we wanted to talk about. It was up in Beverly, Massachusetts, moved to Mystic, Connecticut, released near Block Island, went back up to Beverly, came back down and ended up in Delaware Bay. And that's all within less than a year. So they're moving around a lot more, and we're trying to get a better understanding of what they're doing. And we thought that that was not just seals, because then we looked at sea turtles, working with our partners in sea turtle recovery and Mystic Aquarium. We were able to put some satellite tags on, but because of the size of the animals we're getting, acoustic tags seem to be a better option right now. So we were able to put a couple of acoustic tags on, and we don't get any tag returns because we don't talk to you guys enough. So in case you haven't realized, we're going to start doing that. So sorry for you guys. But this, wow, that's pretty good. You'll see why. But this is giving you an idea of where these animals are going just with a small sample size. What I find interesting and really demoralizing when I come here and talk to you guys about tagging, you're talking about hundreds of animals. I'm talking about 15 or 16, but that's a big day for me. So you can look at this. You can see some of these. These are the loggerheads on the left-hand side, and we have some Ridleys and loggerheads, but they're all going through these arrays. If we can put out some tags, then we can get some better understanding of where we're going. Wow. That's.
Someone have a question? Yeah. Everyone's scared to push us to the end of the day. Thanks so much.
Thank you, sir.
Okay, our next speaker is Matt Davis from the Department of Marine Resources.
Thank you.
I can introduce myself. Howdy, folks. My name is Matt Davis, as Matt said. So I work in the Department of Marine Resources. Today, we're going to talk about a study that we're working on with Wal Gillette at the University of Maine. And that's looking at highly migratory species distribution in the Gulf of Maine as they relate to a proposed offshore wind site. So to kind of set the scene, right, up here in the Gulf of Maine in 2021, in October, the Maine Governor's Energy Office submitted a research application to BOEM for 12 offshore wind floating turbines. This would cover about 9.7 thousand acres, generating somewhere around, I want to say it was like estimated at 144 megawatts. So if we look at this map, we have 3 polygons here to kind of orient yourself if you're unfamiliar with the Gulf of Maine. Portland is pretty much right where those 2 shipping lanes converge on each other. So the green box is the request for competitive interest area that was put out by BOEM. The upside down purple L-shaped polygon, that was originally submitted by the state of Maine as where they were proposing the offshore wind farm to go. But the most relevant and recent polygon that we're paying attention to is that bottom one, which is salmon colored, we'll say. And so that was recently submitted to the state from BOEM. That is sort of what BOEM is saying, hey, this is where we think the best option could be for your research wind site. And the state has 30 days to either accept or reject that and make that the final wind energy research site. That polygon was provided to the state on May 24th, so a very recent thing. So we still don't know exactly where this site is going to be. Now, as the applicant, the state of Maine is going to be working alongside Diamond Offshore Wind, I think is the developer, but they're also going to be conducting some additional survey work to sort of go beyond just what BOEM is requiring. And many of those surveys are being led by the Maine Department of Marine Resources. That's me. So there's quite a bit of different research projects going on here. Just what we're doing is just one small part of that. Obviously, as our title said, you know, we're looking at highly migratory species. So for us, that's pretty much, we have 8 species of shark, but the most common ones you're going to find up there are blue shark, poor beagle. You get threshers, shortfin mako. You'll get white sharks, but they're typically a little bit more inshore. Cool. So this is a pretty, oh my gosh, what time moves so fast. All right, so gotta like refocus now. So Walt and I have been working with commercial and recreational fishermen to sort of understand the fishing scale to which these fisheries operate around sharks and tunas, but also just understanding some of the behavior and distribution of these animals, because relative to a lot of other places in the Eastern Seaboard, sharks and tunas in the Gulf of Maine, there's quite a bit of lack of knowledge, particularly for sharks. So what we've been doing so far is we've been deploying acoustic transmitters, mostly V16s, putting out VR2AR receivers, particularly for bluefin tuna. Walt's been using pop-up satellite archival tags for several years, so we have kind of a backlog of that data. The receiver sites you see here, so those are all of the receiver sites that are on ACT that belong to the DMR. That's 2 major projects. The further offshore grid, those are all passive acoustic monitoring sites with VR2ARs. That's run by our North Atlantic right whale team. That series of receivers that's really close to shore, that's run also by me. That's a white shark study. We don't have any receivers up here right now specific to the offshore wind work and highly migratories, mostly because we're waiting on figuring out where that final lease area is going to be. We're going to deploy about 12 or so of them as soon as we have that area. And then our whale team also has 10 or 12 more that they will also be coordinating with us so that we can get some pretty good spatial coverage. Now, I should say that we are pretty early on in this process, right? We've been tagging for a year or two, and we're several years from turbines even going in the water. But we do have some preliminary results. So two polygons. I like to move when I talk. We got 2 polygons here. The top left one is the proposed research area. The larger one is the final wind energy area for commercial development within the Gulf of Maine. So on the left here, we have some acoustic data. This is just a subsample, the green dots being some of the sharks we've tagged, the red dots being some of the receivers they've been picked up on. We have about 70% of our fish that we're getting returns on so far. We've had some that have come back multiple years, which is exciting. On the left here, these are 2 images that were provided by Walt. The top one shows estimated geolocations of Atlantic bluefin tuna from, I believe those were mini pats. And then on the bottom right, this is a pretty cool one. So this shows Atlantic bluefin tuna observations between 1976 and 2005. Those observations come from commercial fishermen and include spotter pilots, rod and reel fishermen, harpooners, that kind of thing. And the dots represent the size of the bluefin tuna schools that were observed. And to give you an idea of scale, those large dots represent estimates of 1,000 bluefin tuna. So there are some pretty big schools that we've seen throughout the Gulf of Maine. I'm gonna make it. All right. So future plans, obviously, we're gonna continue tagging our fish. You know, right now, I think we have 35 or 40 fish tagged. Just acoustically, that is. We do have some other mini pats that are going out. I think we have between, I want to say 50 to 70 tags in hand that we're hoping to get out this year. We're continuing to work with fishermen to see ways we can incorporate them into this process, expanding our receiver effort. Having that final wind area is going to be really helpful for that. And yeah, I mean, we have a lot of people to thank. Our friendly neighborhood data collectors, or, you know, this project wouldn't be possible without you, wouldn't be possible without, you know, the hard working men and women of the fisheries and the working waterfront in Maine. And yeah, if you want to reach me, you can come talk to me, or you can email me right there. Also, I'm pretty easy to find if you just type Matt Davis DMR. DMR is pretty small, so you'll find me pretty quickly. And if you're curious, that is Bozeman the dog. He is not my dog. But it was submitted to me on our white shark sighting report form. And you can see the white shark kind of there, but I included it because it's a dog and dogs are cool. Yeah, thank you. Questions?
Anyone have a question?
Yeah.
The picture you showed of tuna sightings from 76 to 2005.
Yes.
You mentioned size is tied to, like the size of the dog is tied to school size. Is time indicated anywhere on that?
Not there. No, that is all aggregated.
Okay, cool.
Yep.
Thank you. Great. Our next speaker is Brianna Cahill from Stony Brook University.
Hi, everyone. I'll try to scurry through this quickly, that way we can get to relaxing this afternoon. All right, so as we're all aware, offshore wind energy is growing in prominence along the Atlantic coast, but especially up here in the Northeast. And there's growing concern for how animals are going to respond to this anthropogenic input. More specifically, people are interested in the impact at the wind farm itself, but also in the nearshore environment and the nearby environments, including over the export cable, which runs from land to the wind farm itself. Certain animals, such as sharks, skates, and rays, might be of particular concern, as they have sensory organs on their body known as the ampullae of Lorenzini that allow them to detect electrical stimuli. We in the Peterson lab at Stony Brook University are working on both the Orsted Sunrise Wind and South Fork project, but I'm only going to be focusing on our efforts with Sunrise Wind today. But primarily what we're really interested in looking at is some of the fine scale movements and behaviors around the export cable, both pre and post wind farm construction, but we're currently in the before stage. So we began this study back in July of 2022 with the goals to create a fine scale positioning array around this export cable, which departs the south shore of Long Island, fairly central point of Smith Point. And our array has kind of adjusted over time in shape and size. But we currently have a 7x3 gridded receiver array with an additional 3 receivers in our northernmost row, just to provide a little bit more coverage in that northwest quadrant. All of our receivers that we're using are the Innovacy VR2AR, or the acoustic release receivers. They're housed in acoustic release canisters, either provided by Mooring Systems Inc. or Innovacy, and they're tethered to 150-pound anchors. We're currently contracted by Ørsted to tag 100 individuals for each of the 5 different species, including dusky, sandbar, sand tiger, smooth dogfish, and winter skate. And for the most part, we're pretty on track with where we were hoping to be after 2 years. However, winter skate, we're lacking a little bit. Hoping to put some more effort in this year for that. But so far, of the 300 sharks that we've caught thus far over the summers, we've been able to tag 203 of them. And all of these animals that we catch are receiving a full workup, including measurements. We take blood, muscle biopsies, cloacal swabs, genetics, and then hopefully we're able to put a V16 or a V13 tag in them. And nearly all of our sharks that we've tagged thus far have been tagged on the south shore of Long Island, primarily out of Fire Island Inlet. But since our initial deployment, we've amassed over 4 million detections, and this does not include all of our sync tag detections. And that is accounted for by over 1,100 unique individuals. So more than likely, some of your animals might have been detected at this array. But of the animals that we've been able to tag, we've heard from about 50 of them, and we're really only seeing a summer and early fall presence. We're not really seeing them at our South Shore array very often. Additionally, using the V16 temperature pressure tags, we're able to get an idea on kind of the depth use of each of the species that we've detected so far, where we're seeing a little bit of overlap between sandbars and duskies. And then we're seeing that sand tigers are kind of occupying the whole water column that's available to them, and then smooth dogfish are really only occupying that bottom portion. From there, we took all of our detections, all of our VRL files, and we processed animal positions in-house using Innovacy's Fathom positioning software. Using these positions, we were able to run dynamic Brownian bridge movement models just to start to get an idea on how they're occupying this space. We ended up using the move group package in R, which I really like because it helps to look at the animal track as a whole and then kind of remove some of the space in between, whether that's temporal or spatial. It also allows for you to create individual level rasters for the dynamic Brownian bridge movement models and then stack them to create a species level distribution. Moving from left to right, duskies had a little over 2,700 positions, sandbar sharks a little under 300 positions, and sand tigers a little over 9,000, which goes to show that these animals are using the space differently. Another thing that I would like to point out is that we have had quite a rough time regarding receiver loss in these areas. But as you can see from the sandbar or the sand tiger positions, we're getting really good coverage even outside of our array and throughout the entirety of it. So again, this is just kind of the beginning as we're starting to analyze these data. But yeah, food for thought. With that, wanted to give a huge thank you to our team with the SOFO sharks, the rest of other people at Stony Brook University who have been able to help out, our collaborators at Cornell Cooperative who are looking at horseshoe crab and lobster positions, and then our funding source, Ørsted. Thanks.
Anyone have a question? We have one more speaker that's not on the agenda. No? Okay, great, thank you. And our last lightning talk speaker is Jake O'Donnell. Sorry.
To be fair, I think we were once on the agenda and then we got taken off because my boss, Mary Campfire, was supposed to be here, but then she wasn't. And then I popped us back on. So sorry, I'll go really quick because I would like a beer as well. So I'm going to talk to you about sand tiger sharks on Long Island, which WCS has been tagging for far too long. So we started our project in 2012. Actually, 2011, we tagged a couple of sharks in Delaware Bay, and then quickly realized we wanted to work up here in New York. And so we switched to Great South Bay on Long Island. And then more recently, I've been tagging larger sand tigers out in the Long Island nearshore waters. And we're also doing health assessments all along the way. So like I said, we started in New York in 2012. you might notice that every single year, the number of receivers has gone up or down. The answer is no. We have never had the same array out 2 years, any 2 years. Thanks for asking. So greatly expanded the array up until 2019, and then, or actually, sorry, 2020. And then in 2021, we shifted basically the northern half of our Great South Bay array out to the nearshore waters to look at some of those subadult and adult sand tiger sharks. And these are the things that we were looking at over that time. So what we found is our juvenile sand tigers tagged 82 juveniles from 2012 to 2023, well over 2 million detections. And the vast majority of those, more than 95%, are coming from within marinas along the south shore of Long Island. If you look at those abacus plots, they are basically just straight lines. There'll be 10 to 15 sand tigers, and it's just a straight line for about 6-7 days at a time. So this year we're getting some accelerometer tags to look at sort of what they're doing when they're sitting in those marinas as opposed to outside. Maybe look at if, because all those marinas have giant ferries that come in and run their engines for about half an hour at a time. So see if there's any reaction to that. I imagine not, because they sit there throughout the whole thing. So we see great site fidelity, which I'll talk about in a little while. Interannual site fidelity. There's a lot of annual variability between where the sharks like to hang out. For some reason, so in early part of the study, there were 2 marinas and a natural embayment where the sharks like to hang out, and then later on, still one of those marinas, but the natural embayment in another marina shut off, and they kind of were in a different marina. That might just be kind of a small sample size, a bit of noise there. And so towards like 2018, 2019, shore-based fishery started picking up. Guys were pulling up big sand tigers onto the beach. So we were wondering if some of those were our tag sharks, and the answer was absolutely yes, they were. And so we started in 2021, we put an array of receivers between Frisk Labs, you can see actually on the 2022 map, Frisk Labs, Jones, and Fire Island arrays. Don't ask me about 2021 array, that was a disaster. Started to figure it out in 2022. Last year was fantastic, and we actually did lose one receiver last year, but I refuse to accept that it's gone. I think it just sank, and I'm just hoping for a big storm to scrape off enough muscles that it will float again, because that actually happened with one of those lost receivers in 2022. We recovered it last year, so I'm not putting a red dot on that 2023 map just yet. That's basically what we've learned. That's what they look like now. Designed that high flyer myself. I'm really proud of it. Basically, to answer to address shoaling, we just accepted that shoaling will happen, and we'll bury all of our moorings. So there's just a breakaway length there, both for marine mammal reasons and also when we go out to collect the receivers, all we do is we pull up the high flyer, tie off the line, and just kick the boat into gear. Snaps that weak link, everything comes up, and it works great. So I mentioned site fidelity. Our sharks return for a great number of years after we tag them. This is my last slide, I think. So obviously you see lots of ones and twos there, but that's just because we've actually put out a ton of tags in the past 2 years. So I expect those numbers to start pushing out towards the other end of that scale. We did track a sand tiger for 10 consecutive years, starting in 2014. Got him again last year. Don't expect to hear from him again because that tag is at the end of its battery life, which is sad, but you know, that happens. Oh, sorry, also our health assessment. So yeah, we do we take blood work, feces, fin clips, muscle biopsies. We do ultrasounds when we're out there. We have our vet staff from the New York Aquarium come out and do that all for us, and they know much more about it than I do, so I can't really speak to it very much. And that is about it.